My name is Aiko Nagayama. I'm a research fellow at Ellison Lab at MGH Cancer Center. So my work is about the new antibody-drug conjugate for triple-negative breast cancer. So triple-negative breast cancer is an aggressive type of tumor, and it lacks the estrogen receptors, progesterone receptors, and HER2 amplification as druggable targets. So developing better therapeutics was definitely a clinical unmet need. And we are particularly interested in sacituzumab govitecan, which is one of the antibody-drug conjugate. So sacituzumab govitecan has an anti-TROP2 antigen and a cytotoxic payload called SN38 conjugated by a pH-sensitive linker. So, TROP2 is vastly expressed in the epithelial cancers, so the drug will get internalized into the cancer cells and release the payload and kill the cancer cells. Specifically, will compare to the normal tissue. And the sacituzumab govitecan showed a clinical activity in a phase 2 trial. Our primary interest of our project is to identify the biomarkers of response to the sacituzumab govitecan. So we collected the specimens from patients who participated in the clinical trials for sacituzumab govitecan, and the tumor will be assessed by the whole exome sequencing and RNA sequencing and IHC staining to identify the biomarkers. And the second part of our project is to recapitulate the intrinsic targets that would synergize with the sacituzumab govitecan in triple-negative breast cancer cells. So we decided to employ CRISPR-Cas9 screen to identify the synthetic lethal targets of sacituzumab govitecan. Well, so far we are working on the developmental assays, and there are some CRISPR knockout screening considerations that we made over time. We are hoping to identify the novel targets for synergistic effects by sacituzumab govitecan.